This is Gene Therapy for Hemophilia, Dream or Reality, a show on behalf of the Canadian Hemophilia Society. Here's your host, David Page.
It's a great pleasure to introduce our guest for today's podcast, Dr. David Lillicrap, Professor, Department of Pathology and Molecular Medicine at Queen's University in Kingston, Ontario. Dr. Lillicrap is a Canada Research Chair in Molecular Hemostasis, and he's Director of the Southeastern Ontario Regional Inherited Bleeding Disorders Lifespan Program. Dr. Lillicrap has been conducting research in hemophilia gene therapy for 4 decades, we'll see, and is world-renowned. Today, he will describe the results of clinical trials in gene therapy for hemophilia A and B. Welcome, Dr. Lillicrap.
David, it's wonderful to see you again. And indeed, I have been working around gene therapy, principally in preclinical studies now for over 30 years. So as you can imagine, I'm really happy to contribute to this webcast.
Dr. Lillicrap, before we talk about the levels of factor VIII and IX activity that might be achieved with gene therapy, what levels of factor VIII and IX activity are normally needed to protect people from bleeding?
So as the people listening to this podcast will know, we classify hemophilia into 3 categories: severe, moderate, and mild disease. Although increasingly people talk about severe and non-severe disease. To be severe, you have no detectable factor VIII or IX in the blood. And then from that point on, the disease is milder. Spontaneous bleeds tend not to occur once you reach about 10% levels. There is a little bit of debate about that, but if we could achieve gene therapy with constant levels of between 10 and 20%, the number of bleeding events that would occur would be very, very drastically reduced. So spontaneous bleeding levels, 10 to 20% would be excellent.
So factor VIII and IX levels rise soon after administration of gene therapy. Can you tell us how quickly they rise and how soon after administration that levels stabilize?
You begin to see the levels rising from the gene therapy after about 2 to 4 weeks. So after 1 month, you should be able to take a blood sample and measure the level of factor VIII or IX in the regular way that you do with factor VIII replacement therapy after factor VIII infusions. So initially at around about a month, and then the levels continue to increase to about 6 months. And then what happens is different between factor VIII and IX. What we've seen is that the factor IX levels, once they reach a level at about 3 to 6 months, that level stays persistent out to a number of years, at least to 3 or 4 years and maybe beyond that. But with factor VIII, the levels start to fall at about 6 months, and they fall by about 50% over the next 2 years, and then plateau at a much lower levels. So let's say you reach a peak level of 50%. 2 years out, that will be more likely down to about 20-25%. And then maybe slight falling off a little bit beyond that. So quite different between factor VIII and IX.
We'll come back to that in some more detail in a minute. Do all people have the same response?
There is significant variability between patients. Some patients, a very small number, about 5%, do not respond. So they have no production of factor VIII or IX. But the majority have some response, but that response can go from 10% levels to 150%, or maybe even sometimes higher. And the problem is that at the moment for individual patients, we have no way of predicting where you will be in that large range of factor levels. So the unpredictability is an issue which the field is trying to resolve, but we're not close, I think, to being able to do that currently.
And do we understand why this happens, why there's a different response in different people?
I think gene therapy is still very complicated. It's taken us 35 years to reach this point, but there are still many unanswered questions, partly to do with the therapy itself, and then partly to deal with the individual genetics and other aspects of individual patients. And so the answer to your question is probably going to be a sort of a multifactorial equation, which I probably would not understand and certainly couldn't explain.
We'll come back to the question of prophylaxis following gene therapy in a minute. I want to come back to the question of durability that you mentioned, that especially in factor VIII, factor levels don't seem to be stable. As you say, we have to distinguish hemophilia A and hemophilia B. Could you give us more detail about how high people get in terms of their factor levels, how quickly they respond, and how long these levels might last, and distinguishing A from B.
Yeah, so let's talk about hemophilia A first, where you would start to see factor VIII in the blood at about 2 to 4 weeks. The levels would rise to a peak at about 6 months. An average level would be between about 40 and 50%. Now, there are some people who would have much higher levels, and remember, a small percentage, less than 10%, who may have almost no response. Let's take the person with 50% levels at 6 months. Within 2 years, that level may be down to about 20%. And then the fall off seems to be much slower. We've got patients now out to 6 years. So in terms of durability, 6 years of expression. And those levels are between about 10 and 20%. So there's certainly levels that we know from natural history will prevent spontaneous bleeding, but there may be breakthrough bleeds, particularly with activity or trauma or other things. So that's the factor VIII story. The factor IX story is different. Again, you'd expect to see levels beginning at about 2 to 4 weeks. They may peak somewhere around 3 to 4 months, and then they seem to stabilize, going out now to the longest human patients are out to 12 years or so, with levels that really haven't changed much since about year 2 or 3 after gene therapy delivery. Oh, and I should say actually one other quick word about hemophilia A, because some people listening may be quite disappointed. But as you know, we have hemophilia dogs here in Kingston, and some of those dogs, after single treatments, had persistent factor VIII, albeit at lowish levels, out to 12 years. So the durability, honestly, even factor VIII may be quite long, over a decade.
So in the case of dogs, that was their entire lives, really?
Yes, they had lifelong levels of protected factor VIII, albeit the levels were between 5 and 10%. They weren't very high, but they were protective of spontaneous bleeding.
So this, in many cases, would be enough to keep people off factor VIII or IX prophylaxis?
Yeah, so the clinical trials, which have included large numbers of patients now, show that the use of prophylactic infusions is massively reduced. So down to probably no more than 4 or 5 injections per year, I would say. I mean, there is a massive reduction in the use of factor infusions.
Is there any data on how many people have gone back to prophylaxis after gene therapy?
There is beginning to be that sort of data, and it's a little variable. I mean, almost none of the factor IX treated patients have gone back to prophylaxis, but some of the factor VIII patients have. Remember, a very small number, the gene therapy doesn't work, so they need to return to their regular prophylaxis. And then some of the patients are maybe treating themselves around sporting activities or other situations where they may expect to bleed more.
Is prophylaxis with factor VIII or IX just as effective if they've had gene therapy and had to return to prophylaxis?
Yeah, so one of the important things to keep in mind is if the gene therapy doesn't work, or if you need a little bit more, suppose your post-gene therapy level is 15%, but you want to do something more strenuous, you can treat yourself in just the same way that you would have done before. So there are no drawbacks from being a gene therapy failure or needing to top up the gene therapy.
Is there an explanation why levels seem more stable in hemophilia B than in hemophilia A?
The simple answer is that the cells that we deliver the gene therapy to are used to making factor IX. So normally factor IX is made in persons who do not have hemophilia in the liver cells. And so we think that the liver cells are much more comfortable in making factor IX over a long period of time. Those liver cells do not, in normal circumstances, make factor VIII. And so part of the mystery, we believe, is that making those liver cells make a protein that is not naturally made, over time, they get stressed. They undergo this thing called cellular stress, and this reduces the levels over the first 2 or 3 years after gene therapy is delivered.
10 years ago, did you think hemophilia gene therapy would achieve levels like the ones we're talking about?
I think you know me well enough to know that I'm the ultimate optimist. So I always believe that gene therapy would work. In fact, a number of years ago, we published a manuscript saying that gene therapy was such an ideal treatment for hemophilia that if it failed in hemophilia, gene therapy would never work. I'm not surprised. However, having said that, I think the recent advances, the speed of recent advances has been quite remarkable, particularly for factor IX, which currently now looks as though this could be long-term curative. It could well be that the patients who receive factor IX gene therapy may not need to treat themselves with factor IX for a very, very long period of time. Factor VIII is always more difficult, so we'll have to see.
So this has been a dream for the last 30 years. It's been your dream for the last 30 years, I mean, ever since those factor VIII and IX genes were cloned. For whom do you think it's now a reality?
I think it's now a reality for anyone who's interested in a different way of being treated. If you do not wish to think about your hemophilia, if you could lead a hemophilia-free existence in your head and your body, gene therapy can be that answer. It may not be the way that everyone wants to treat themselves initially. Maybe people inevitably will be more cautious, but some of the results, particularly in factor IX, are looking very, very encouraging for long-term absence of injections of any sort. And so that is really a remarkable advance in the treatment of this disease.
Though I think we have to say it's not for everybody at the moment.
We don't expect more than maybe 10 or 15% of patients to be interested in this. So I think there is no thought on the part of treaters that suddenly people are going to keep saying, I want gene therapy now. But there will for a certain type of patient who doesn't want any sort of prophylaxis because it's a hassle. It's always on your mind about when you give yourself prophylaxis. If you want to free yourself from that, gene therapy should be considered very carefully and is an option.
But again, there are some restrictions in terms of eligibility.
Yeah, so the main things about eligibility are, first of all, you have to undergo immune testing to see whether you have antibodies to the viral vector. And unfortunately, that will eliminate somewhere in the region of 60%, 70% of patients. So even if you want gene therapy, you have to pass this screening test, first of all. That will be the first thing. There's no gene therapy for individuals below the age of 18 at the moment, although that's likely to drop to the age of 12 relatively soon, I think. If you have an inhibitor or you've had a past history of an inhibitor, you're also not eligible. So those are the principal things at the moment, which means that not everyone is eligible.
Before we end, is there anything else you'd like our listeners to know?
I think the only other message I'd like to give is that the reason that we've been able to make these advances is due to the village of hemophilia, the community of hemophilia, the patients, all the healthcare workers, and the scientists. This is in part why I think hemophilia was an ideal target, because this community is extremely well informed. All of the groups of individuals as patients, treaters, and scientists work together. And that's why we've been able to make these advances, which now potentially are available to patients who might be considering a different way of treating their disease.
Thank you very much, Dr. Lillicrap. We really appreciate your research work, but also your contributions to informing the community.
For more information, we invite you to check out more episodes in this series, Hemophilia Gene Therapy, Dream or Reality. This podcast series was made possible by an unrestricted educational grant from Pfizer Canada to the Canadian Hemophilia Society.